all, thank you for coming. My name is Emilie Richards, and today I just want to share with you the start of my investigations into the different sources of genetic variation that have contributed to this radiation of Caribbean pupfishes. Now, a growing number of studies have documented extensive histories of hybridization and the exchange of genetic variants in many radiating lineages. Some well-known examples including the heliconius butterflies, the African cradle lake cichlids, and Darwin's finches, suggesting that gene flow may be an important mechanism for rapid diversification. However, it's still not well known how often hybridization or gene flow is necessary for radiations or simply being pervasive throughout the history of many radiating groups. So I think an excellent system for investigating the potential for gene flow in rapid diversification and the evolution of complex phenotypes exists in the cyprinodon pupfishes. So throughout this highlighted range, cyprinodons are mainly allopatric generalist species, and sympatric radiations of trophic specialists have evolved only twice. Once in a lake in the Yucatan, and once in the radiation I'll be focusing on San Salvador Island. Now on San Salvador, we have our generalist pupfish eating algae and microinvertebrates, and living sympatrically with it are two specialists, so our scale eater and our molluscivore. Now the primary axis of diversification in this system is in craniofacial features, particularly the oral jaws. Highlighted in the CT scan in colors. Now our scale eater has much more enlarged oral jaws in comparison to our generalist, and our molluscivore has shorter and thicker oral jaws, as well as a novel nasal protrusion that we think aids in stabilizing prey for crushing their hard shells. Now despite apparently equivalent ecological opportunity across the Caribbean for these specialists, we only find them on San Salvador Island. And like those previous studies I mentioned, cyprinodons have, in cyprinodons there's been documented extensive Caribbean-wide gene flow. So each of these arrows indicates an admixture event with our San Salvador species in bold. And genome-wide, there appears to be no unique signature of gene flow into San Salvador that would suggest that gene flow is a cause of the radiation only being there. However, previous work done by my lab mate Joe McGurk has found that genetic divergence is limited to very small regions of the genome among these species. So only hundreds to thousands out of 12 million SNPs that we found were fixed between species, suggesting that we might want to look for unique patterns of gene flow on a much finer scale than genome-wide. So that's the work I want to share with you today. Particularly one thing, the fine scale signatures of adaptive integration I found using sliding window statistics, as well as some complementary data with fine-scale variation and evolutionary relationships across the genome I found with the machine learning approach. And then talk about how some of these identified candidate adaptive integration regions have potentially contributed to the complex phenotypes we see in the specialists, with a focus on a region in the gene called SKI as a case study. And then end with future research I'd like to do to address the question of whether these adaptive integration regions I found potentially trigger the radiation. Okay, so the data set we're working with is whole genomes from 42 pupfishes consisting of about 16 million SNPs. The bulk of the sampling being done on individuals from San Salvador, but also including a generalist population from Nassau and another island in the Bahamas, as well as one from the Dominican Republic, and then some more distantly related outgroups. So let's go to the first line of evidence for fine-scale signatures of adaptive integration. So to do this, I calculated a formal test statistic of integration called the F4 statistic. And if you're familiar with the D statistic or the ABBA test, this is very similar. You're simply looking for deviations from the relationships you'd expect without integration. So for example, we'd expect that the scale eater and molluscivores should look more similar to each other than, or genetically similar to each other than Nassau or Dominican Republican population. If there has been integration, say between the molluscivores and Nassau, they will be most closely related, and our F4 value will deviate away from 0. So when we do this across the genome, what we find is that very little of it appears to be integrous material, so less than 1%. And if we look for signatures of selective sweeps in those regions, so SNPs fixed between species, and then signatures of positive selection, we find that adaptive integration appears to be very rare and localized in the genome. So it represents less than 1/100% of our genome. Okay, and then to complement this, I also looked for fine-scale variation in evolutionary relationships across the genome using a machine learning approach called saguaro. Now, saguaro can be used to determine phylogenetic variation across the genome without any a priori hypotheses about what that variation should look like and kind of what scale it should occur across the genome. And then an oversimplified representation of it, saguaro does this by first generating a tree for the entire genome, and then using a hidden Markov model, searches the genome for regions that fit and don't fit well with that tree, and then from those poor fitting regions, uses a self-organizing map to cluster them by similarity, and then takes that worst fitting cluster, that's the biggest, and generates a new tree, and then goes through this whole process again. And you can do this many times until you have a genome that's partitioned into different regions that vary in evolutionary relatedness. Okay. So when we do this across our genome, we found that the predominant tree does kind of what we expect. So expected close relationships of the groups on San Salvador, with increasingly distant genetic relationships based on geographic distribution. And that tree represents about 64% of the genome. However, in smaller regions of the genome that are spread out, we find trees that diverge from this, so divergent topologies, including ones where, say, our scale eaters are separated from the rest of San Salvador by our outgroups, and this represents about 4% of our genome. And we find a similar pattern for our molluscivores too, where they're separated again from the rest of San Salvador by the outgroups. And in fact, these kind of trees, these divergent topologies, are what we find in our adaptive integration regions from that sliding window test. Okay. And then so next I'd just like to talk about some of the candidate adaptive integressions and how they potentially contributed to the radiation. So in our top, so adaptive integration appears to be rare in the genome, but it appears to be important to that main axis of diversification, our oral jaws. So the top four regions, so 4 of the top candidates are regions that have been associated with craniofacial features in mice or zebrafish. Those are highlighted in red. And then the starred are regions that have been associated with jaw size in pupfishes, and that work was done by my lab mate, Jill. Okay, so if we take a look at one of the more interesting candidate regions, we see a peak in our F4 statistic. And this is a signature of integration right here, and it's a signature in a region of a gene called SKI. Now in this, this region is a cluster of those divergent topologies as well. We also find high divergence between our specialists in this region, so a peak in FST, and to complement that, also GXY, so absolute genetic divergence. And to complement that signature of integration, we find low divergence between NASA and our molluscivores in this region. And this region appears to be under a selective sweep in the molluscivores. So this dashed green line is high in our molluscivores, so there's low genetic diversity within molluscivores in this region, and also negative estimates of genus D, a signature of positive selection. So if we make a population graph for our groups and infer admixture events across it, one of the admixture events inferred is integration from NASA into our molluscivores. So the integration region we find appears to be from NASA into our molluscivores. And in fact, we see very similar genotypes between NASA and the molluscivores across this region. So here's just an example of one of the fixed SNPs. In our region, so there's 3 fixed SNPs in that integress region, 1 in a 3' UTR, 1 in a third position of a codon, and 1 in an intron. So this is our 3' UTR fixed SNP, highlighted in orange position here. And across the top is our NASA sequence, the gray bar, and then any difference between the NASA sequence and any other sequences is highlighted in black. And so what should stand out is that our molluscivores, so all these green bars, are very genetically similar to NASA in this region. And if we assume that the differences we see between NASA and the molluscivore have accumulated mainly neutrally, we can get a very rough, emphasis on rough, estimate of the timing of divergence between these two in the integration. So when we do this, we can get approximately 5 to 25,000 years ago, which is cool because it overlaps with the estimate of the origin of the radiation about 10,000 years ago, based on the last time sea levels were lowered and the lakes would have been dried on some Salvador. Now, introgression in this particular region in this gene is actually really cool because ski is a proto-oncogene with strong craniofacial effects documented in both mice and zebrafish. And mutations in ski cause phenotypes in mice that are really remarkably similar to what we see in our molluscivores. So for instance, we have a depressed nasal bridge in mutations in ski and mice, and it's very similar to what we see in our molluscivores in comparison to our generalists. So we find that we're finding adaptive integration signatures in regions that are associated with this primary axis of diversification,